Hello, my name is Stuart Masterson and I'm from the University of Kent, and I'm presenting a poster on the topic, is the Bombali virus pathogenic in humans? So a bit of background. So the Bombali virus is a filovirus, much like the Ebola viruses and Marburg viruses that have caused the severe Ebola virus disease outbreak. So the Bombali virus was recently discovered in 2018 in a region in Sierra Leone, and part of this research is to determine whether or not it is pathogenic or not, because it's currently unknown whether it induces human disease. So previous research was carried out to understand what makes Ebola virus pathogenic, and here there are, before Bombali virus, there were 5 known Ebola viruses. There was Zaire Ebola virus, Sudan, Bundibugyo, typhus, and these were all pathogenic and caused disease in humans. And that was compared to Reston virus, which is circulating in Asia, which is non-pathogenic in humans and doesn't cause disease. So previous research was carried out into understanding what causes this change in pathogenicity. And here this project expands on that and uses a greater data set, and also looks at the Bombali virus to determine whether or not it could be pathogenic or not. So the way we characterize this is through the use of things called specificity determining positions, or SDPs. And so that is a position that is highly conserved within one species but differentiate between other species at the amino acid level. And so what we studied was whether these SDPs between a data set of the pathogenic viruses and Reston virus had any effect on pathogenicity, and then we looked at Bombali virus as well. So we had 1,408 amino acid sequences and genome sequences from all 5 Ebola virus species, of which about 27 were Reston, and the others were the pathogenic species. And we used computational approaches to identify SDPs between these 2 datasets. And what we found were 166 SDPs, which was a slight reduction of 180 from the original project. These were spread all across all 7 proteins, and then we used structural data to determine whether any of these SDPs would have some impact on pathogenicity. And of particular importance was the VP24 protein, which is the Ebola VP24 protein is involved in inhibiting the interferon response by its interactions with host Characterization alpha. And what we found was that in the key VP24 Characterization alpha binding region, there were 5 SDPs that differentiated the pathogenic Ebola viruses from the non-pathogenic Reston virus. And changes to these amino acids had some impact in the interferon response by the interactions with Characterization alpha. We could map 92 of the 166 SDPs in total, and of all the 92 that we studied, most of them didn't seem to have any major impact on protein structure or function or interactions. However, they were studied in isolation, so there may be some compounding effect that we weren't aware of. But generally, the main results pointed to this KPNA5 region, which was also highlighted out in the previous study. So what we can say is that our methodology is robust even when a massive increase in sequences is used. And then once we had that data, we wanted to look at the Bombali virus and see how it relates to that region. So looking at the carrier-fearing region in particularly, there are two specific residues, 136 and 139, that actually agree with the restin virus amino acid. And these two positions were considered key to pathogenicity. And so that suggestion is that the Bombali virus may not be pathogenic because it may have the same interactions with the carrier-fearing protein as the restin virus. This is despite the overall protein sequence being much more closely associated with the pathogenic Ebola viruses in Bombali virus than it was with the restin virus. Bombali viruses are about 59% similar to the pathogenic Ebola virus species. However, when we just look at just the SDPs that we identified, there was about a 70% similarity between the Bombali virus and the pathogenic Ebola viruses, which suggests that there is a higher conservation in those regions, and that Bombali virus is more like the pathogenic viruses, except for these key residue positions where it's much more like restin virus. So overall, we concluded that Bombali virus may not be pathogenic in humans because of these key residue positions. However, it may have the potential to become pathogenic through mutations because it is highly similar to the pathogenic viruses in other aspects. Thank you very much.